To test the presence of glucose, starch, proteins, and fats in suitable plant and animal materials. Foodstuffs generally have carbohydrates, proteins, fats, and lipids as basic components. Food is needed by all living organisms. Breakdown of food provides energy and raw materials for growth and development.
Madam, what are the sources of carbohydrates, proteins, and fats?
Carbohydrate sources: cereals, potato, sugar, bread. Protein sources: milk, cheese, white part of the egg, pulses, nuts. Fats sources: butter, ghee, meat, milk, fish, groundnut, coconut, mustard oil. Let's do an experiment to test the presence of glucose, starch, proteins and fats in suitable plant and animal materials. Our aim here is to test the presence of glucose, starch, proteins and fats in suitable plant and animal materials. The materials required are seeds of gram, peanut, potato, beetroot, cane sugar, rice, wheat, etc., test tube stand, test tube holder, spirit lamp, watch glasses, petri dishes, Benedict solution, Felling solution A, Felling solution B, dilute HCl, 5% NaOH solution, iodine solution, copper sulfate solution, Millions reagent, ethanol, Sudan 3-di. Procedure and observations. Take peanut or gram seeds, rice, wheat, pulses, groundnut, potato, beetroot and prepare their paste separately. If dry, soak them in water, grind them and extract their juice. Take juice of sugar cane. Filter the juice and use the filtrate for testing the presence of carbohydrates, starch, sugars like glucose, sucrose, fats and proteins. Test for glucose or reducing sugars. Experiment Benedict's test. Take 2 ml of food juice in a test tube. Add 2 ml of Benedict solution and heat. Observation: the solution first shows green color, precipitate, which later turns brown and finally orange. Inference: reducing sugar present, green precipitate indicates less sugar, orange precipitate confirms large amount of sugar. Felling's test. Take 2 ml of Felling solution A, 2 ml of Felling solution B and 4 ml of juice or filtrate. Heat them. Orange colored precipitate appears. Reducing sugar present, glucose, fructose, etc. Test for sucrose. Experiment. Take 2 ml of food. To this, add 2 drops of dilute HCl. Boil it and add 3 drops of NaOH solution. Observation: orange colored precipitate appears. Inference: sucrose is present. Perform Benedict's test as mentioned for glucose test. Violet color appears. Sucrose is present. Test for starch. Experiment: take rice, wheat or potato filtrate. Add 3 to 4 drops of iodine solution. Observation: blue-black color is seen. Inference: starch is present. Take any of the above starch solution, 2 ml and 2 to 3 drops of dilute HCl and boil. Add 3 to 4 drops of NaOH solution and perform Benedict test. Orange precipitate appears. On hydrolysis with acid, starch breaks down to monosaccharide, glucose sugar and gives positive test with Benedict solution. Test for fats. Experiment grease test. On a piece of paper, crush some groundnuts or almonds. Observation: translucent spot appears. Inference: it indicates presence of fat. Take 2 ml of any oil, soya bean, groundnut, etc. To this add 2 ml of water in a test tube. Shake well. 2 distinct layers appear with oil floating on the top. Emulsification occurs as oil layer forms small droplets in water on shaking. Fats are present. Sudan 3 dye. Test. Take 1 ml of Sudan 3 dye and 2 ml of oil containing food stuff. Pink colored droplets appear. Fats are present. Test for proteins. Foodstuffs like egg albumin, milk, pulses are rich sources of proteins. These give positive test for proteins when tested. Experiment. Biuret test. Take 2 ml of food material and add 3 ml of biuret reagent, that is, a solution of 5% NaOH and 2 drops of 1% CuSO4 solution. Observation. Violet color appears. Inference. Proteins are present. Xanthoprotein test. Take 2 ml of food solution and add 4 to 5 drops of concentrated HNO3. Yellow precipitate appears. Proteins are present. To the yellow precipitate, add 1 ml of concentrated ammonium hydroxide solution. Yellow precipitate changes to orange. Proteins confirmed. Millen's test. Take 2 ml of food solution and add 2 ml of Millen's reagent. Reddish orange color appears. Proteins are present. Observations. Serial number, food stuff, fat, protein, starch, sucrose, fructose or glucose, wheat flour, sugarcane juice, grape juice, groundnut, milk, potato, pulses, legumes. Precautions. Dry foodstuffs like pulses, seeds, etc. must be soaked in water before grinding. Fine grinding must be done to make the homogeneous paste or to get filtrate. Use acids carefully. While heating the test tubes, continuous heating of the bottom must be avoided. Use only hard glass test tube for boiling purpose. Use freshly prepared reagents. Add only few drops of iodine solution.
Thank you, ma'am, for this knowledgeable experiment.